Hello, everyone. My name is Sarah Lebeer. I'm a professor of microbiology and biotechnology at the University of Antwerp in Belgium. And I'm also an academic board member of ISAPP. I have the honor to introduce you or to welcome you at the second day of our annual meeting. Unfortunately, due to the COVID pandemic, we had to organize it in a virtual way. Yet, we are all scientists and microbiologists. It's also a very interesting era. Microbes are continuously being put in a negative light, but I should not convince this audience that we can also use microbes to improve our health. And positive as we are, we want to focus today on possible solutions with probiotics and vaccines. Just after 1 year, after our lifetime ISAPP scientists Glenn Gibson and Gregor Reed have co-authored an opinion paper on using probiotics to flatten the curve of COVID-19 in Frontiers in Public Health, but also in the Lancet, they were co-authors. It's now actually time, I think, to show some actual data and interesting research that is going on. So it's my pleasure to introduce you our first speaker, Professor Paul Wischmeyer. He's a critical care and perioperative physician and a nutrition physician. He's also serving as a professor of anesthesiology and surgery at the Duke University School of Medicine. And he's an outstanding scientist. I think he has won numerous awards. He's also an advocate to keep care as the focus of healthcare. And I think for this audience, it's important to mention that he does a lot of research on the microbiome and probiotics. And he was just also one of the first scientists to start a probiotic COVID-19 trial, as you could have read in our ISA blog post from last year, May 2020. So Paul, it's now a real honor to have you speaking here and to introduce you. We are all looking forward to your talk. I just want to give one final remark. Please type your questions in the bottom at your left in the platform where you can find Ask a Question. So we'll have, after this presentation, we will have a Q&A session. Paul, the floor is yours.
Hello, it's great to be with all of you here today to talk about the key role of probiotics in the prevention and treatment of COVID-19 and other infections. Quickly, some conflicts or what I would like to call alignments of interest, because I would like to believe all of the funding agencies that I work with that support our research are in alignment with our interests to improve patient outcomes, and that we're actually not in conflict, but we all have the same goals to improve our patients' outcomes as we perform the research that we are engaged in. So we are poised to answer key questions about our microbial selves and begin to use this data to help us learn how to improve health from both staying healthy and also preventing infection. And so can we change the microbiome, perhaps with probiotics or prebiotics, to treat and cure diseases, perhaps like COVID-19? And I think some of this comes back to what's in our patients' guts and how can it help or hurt our patients with illness and in prevention of risk of illness. And I work, as I said, I'm an ICU physician at Duke, and my patients have many things when they're in the ICU with us, including when they have COVID-19, that contribute to major negative changes in their bacterial or microbial makeup. We call this dysbiosis. And clearly how we feed them, the antibiotics we give them, the sedatives and the opiates we give them. Opiates make bacteria more aggressive, phosphorus levels and obesity, all these things contribute to dysbiosis or an adverse microbiome makeup or adverse bacterial makeup. Psychological stress does as well. So all these things contribute to potentially our ability to recover from illness, but also we believe our risk to getting illness, including getting illnesses like COVID-19. And so stress can clearly put us at greater risk. And so I think it's critical that we restore balance to our patients, especially our patients that are critically ill, like the ones who with COVID-19 that I care for in my ICU, but any illness, even just getting antibiotics, will disturb our microbiome and change who we are as humans and our resistance to illness, we believe. And so giving things like probiotics, we believe, can improve that. One way to think of it is clearly illness and antibiotics and COVID-19 lead to a blighting of the normal microbes in our body, in our guts, in our mouth, and in our skin. A little bit like the blighting of a lawn in a drought, perhaps. So in fact, we need to resod this lawn after illness, and perhaps after sort of some of the normal things that have involved what the Western diet and Western medicine have become, all the antibiotic use, all of the sort of processed foods and changes in cleanliness, all have contributed to changes that set us up for infections like COVID-19. And perhaps we need to be restoring this lawn both after illness to recover patients and before illness as well. And what's the science for this? Well, I trained with John Alverdi. He may be a name you know. He discovered quorum sensing shortly after I was in the same lab of Eugene Chang's at University of Chicago. And we've discovered some amazing things. All of us right now have pathogenic Pseudomonas, or pathogens living inside our gut, acting sort of not doing any damage, acting quietly, waiting for our body to be stressed. And they can sense our stress and talk to each other. And that's when we lose our commensal flora, and the bacteria, the pathogens, sense that stress. It's a little bit like when a hurricane hits a city like New Orleans and all the people evacuate, all the regular inhabitants evacuate. Well, then looters come in and bring their looter friends, and they begin to loot and do damage. Well, that's what happens when our gut and our body get stressed. The normal bacterial flora disappear, and these pathogens bring their pathogen friends and begin to attack us. They sense our stress. And the reality we've discovered is if we put the commensal flora back by giving probiotics, we can calm the pathogenic bacteria, and we can reduce the stress from both bacteria and viruses and their ability to infect us. And so it's a little bit like when all the normal inhabitants of New Orleans return after a hurricane, the looters go away. And it's the same when we give a probiotic or commensal flora back to our patients. And any commensal flora, any probiotic we're finding, or many of them that outgrow pathogens, will cause this protection against these pathogenic bacteria and viruses. And so there's a lot of ways probiotics protect us. We know they directly inhibit bacterial and viral adhesion and its ability to infect us. They secrete antiviral and antibacterial defensins. They improve the mucus layer that protects our lung and our gut to keep the bacteria and viruses from being able to infect us. They do a lot of beneficial things, including effects on the immune system. Prebiotics do the same as well. Prebiotics are a vital source of fiber for things like short-chain fatty acids, which are essential to reducing intestinal inflammation, improving Treg cells, which reduces organ injury, which we'll talk about later. They do a lot of beneficial things for our body. And when we lose these prebiotics, our gut is at risk, bacteria can invade, and they can cause all kinds of downstream injuries and infections. And so we're really learning a lot about the science, and my laboratory has been very passionate about studying the science of how probiotics protect us against infection. And so some of our first set of experiments, we're looking at Lactobacillus GG and other probiotics and its ability to protect us against peritonitis, like ruptured appendicitis, perhaps. If we give probiotics at the time of the start of ruptured appendicitis or peritonitis, it's very protective against death in these animals. It prevents the animals from dying from these infections. And what we found is it directly prevents bacteria from escaping from the gut and other places into the blood and causing sepsis and bacteremia. So these are animals that had ruptured appendicitis or peritonitis, and this is smears of their blood. And so you can see all the bacteria that escape into the blood in the animals that don't get probiotics. If you look at the animals who did get probiotics before peritonitis, they have no bacteria in the blood. And we repeated this experiment many times. We couldn't believe the dramatic benefit and protection that Lactobacillus GG and some other probiotics had. And they prevent the downstream lung injury, ultimately, that these infections cause. You can see the septic animals have all kinds of inflammation and edema and injury in that first panel at the bottom. And the animals that got probiotics do not. So it protects the organs from injury. It reduces inflammatory markers in the lung as well. So giving probiotics to the gut reduces inflammation and inflammation markers like TLL2 and others in the lung. So downstream organs are affected by what we give the gut. And this is true for inflammatory cytokines like TNF-alpha and IL-6, which produce a lot of the lung injury we see in infections and ARDS, and even in perhaps COVID-19. Again, probiotics reduce that lung injury that leads to the complications of infections like COVID-19 or other bacterial infections. What about study of direct respiratory infections and pneumonias? We've studied that as well. We've looked at Pseudomonas, a very aggressive pathogen, and what happens if you give Pseudomonas into the trachea of an animal and then give it probiotics at the same time. And what we found is giving probiotics like Lactobacillus GG can directly protect against death from severe insults from pneumonia. COVID-19, again, being a pneumonia, Pseudomonas is another aggressive pneumonia. And again, we found the same benefit, that the animals that got pneumonia without getting a probiotic have bacteria that escape their lung into their blood, causing all kinds of organ injury and death and organ failure. The animals that got probiotics had no bacteria escape into the blood. It protects the body from the invasion of these bacteria into other organs and into the blood. And again, it protects the lung quite dramatically as well. The other thing it does is it benefits how our body responds to inflammation. We know that a strong T regulatory cell response is correlated with resistance to and survival from invasive pneumonias. COVID-19 is one of those, bacterial pneumonias are another. So you want this good T regulatory cell response to modulate or reduce, modify the inflammation that's injuring the lung ultimately. And we found that giving Lactobacillus GG and other probiotics at the time of pneumonia increase these protective Treg cells in the lung. And it's giving, again, the probiotic to the gut, like you take it by mouth, and then protective responses from immune cells occur that prevent the inflammatory injury that we know leads to the severe lung injury of many diseases, including diseases like COVID-19. And so again, basic science is a start, but it's not sufficient. I think another key step in using probiotics more routinely in our care is they need to be safe. And the government, a number of years ago, AHRQ, went looking for signals of harm from probiotics, expecting, I think, to find them. And they looked at many, many studies. 622 studies were looked at of probiotics of any kind in healthy and sick individuals. And they found no increased risk of adverse events or infections from probiotics. So if you're going to give bacteria to people, people worry that you might get infections from them. That's not been the case in any of these studies, that's been a consistent signal. There's been no adverse effects. And in fact, if you already were sick when you received probiotics, there was a trend, a meaningful trend towards protection against infections and other adverse events. So again, when you're sick, it appears probiotics actually are safer or protective to give, perhaps, versus creating risk to the patients. And they concluded that in 622 trials, there was not an increased risk of infection or other complications from probiotics being used in both healthy folks and patients with illnesses. So again, appears to be very safe. So now what happens to the microbiome in critically ill patients, like patients who get very sick with COVID-19? So we became very interested in that. And we did a study actually with Rob Knight and his group looking at what's in the guts of patients in the ICU. It's called the ICU Microbiome Project. And again, we published our first paper not so long ago, and we looked at 115 ICU patients in 4 centers around the US and Canada. All of these patients were on ventilators. Unfortunately, all patients on ventilators in the US and Canada for more than 2 days get antibiotics. And so that was part of their care. And as you can see, the fecal microbiome of an ICU patient is significantly disturbed and is quite different from healthy patients. And so you can see the ICU patients in blue are distinctly different in their gut microbiome makeup than the American gut, which is a healthy control group that Rob Knight's group collected. It's very different than healthy folks are. And in fact, specific families of protective bacterial families are lost. So you can see the big dots are the ICU patients, and the white dots represent loss of the Firmicutes family. And so whole families of protective bacteria are lost very quickly when patients become critically ill, a little bit like in New Orleans where all the normal inhabitants leave. And you can see the dots are much redder in the normal healthy folks that are the small dots. And so we lose whole families of beneficial protective bacteria. The other problem that is, normally there's quite a distance of difference, or quite a difference between the gut, the mouth, and the skin. Our microbiomes in those 3 areas of our body usually are quite distinct. That is lost in critical illness and acute illnesses. You can see the dots begin to coalesce, the big dots of the ICU patients, and they all begin to look the same. So the mouth, the stool, and the skin begins to look the same as we lose diversity and we lose the barriers between those different parts of the body. And you can see another way to represent the data, all of the red, each of these lines representing a patient, all of the red, both admission and at discharge in the ICU patients. That red is pathogens and proteobacteria that represents many of the pathogens in our body. You can see the healthy patients in that bottom graph have very little red. And so they have the normal Firmicutes and Bacteroides species that make up a healthy microbiome, and that's lost very quickly in illness. We found specific bacteria are depleted. These are potential targets for probiotic therapy in illness. Faecalibacterium prausnitzii is a short-chain fatty acid-producing anti-inflammatory bacteria that's lost very rapidly in ICU patients. It's been studied in IBD as a target for treating that illness. It makes short-chain fatty acids, which are vital to protecting the gut. And again, pathogens grow up very quickly and predominate the stool of ICU patients. Things like staph and proteus and other gram negatives grow up very quickly and take over the gut. That's true in the oral microbiome as well. There's loss of healthy bacteria and growth of pathogens like Staph aureus and mycoplasma that grow up very quickly in the ICU patients. So you can see there's large families of pathogens that take over the gut, kind of like the looters in New Orleans after a hurricane take over when the patient becomes sick or when a person becomes sick. And the protective bacterial families like Faecalibacterium and Presnutzii are rapidly depleted. So we lose these, and these are good targets perhaps for probiotic interventions. Diversity is lost quickly as well. You can see the ICU patients there in the red lines have much less diversity in their guts than a healthy group of patients, which is much more diverse. And in fact, there's a crash in diversity. You can see at ICU admission, the most abundant bacterial family makes up about 25% or a quarter of all the bacteria. But within a few days, that one family of pathogens can make up 95%. One bacterial pathogen can make up 95% of all the bacteria in the gut. And all the normal bacterial families are lost in some of these patients within just a few days. So there's a crash in diversity where one species can take over the entire gut in illness, and it's often a pathogen. So some clinical results. We know that the overgrowth of Klebsiella and other bacteria on the skin can be associated with patients who die. We know that bacteremia, or escape of blood into the bacteria into the bloodstream, is associated with mycoplasma growing up in large amounts in the mouth or the oral microbiome area. And acute respiratory distress syndrome, like we see in COVID-19, is also associated with changes in the microbiome in the stool and in other areas of the body. So then the question becomes, is it antibiotics or is it the actual effect of illness that changes the microbiome? So we need to study that. Which is it, the antibiotics or the illness itself that changes our microbiome? So we created a antibiotic pressure scale that was published in Lancet that we adapted for ICU, and we were able to quantitate the amount of antibiotic pressure our patients saw, and we were able to show that the changes in the oral microbiome at ICU discharge seemed to be related to how much antibiotics pressure the patients saw, so how many antibiotics they received and how potent they were. But the stool was not related to antibiotic pressure, and so the changes in the gut seem to be as much perhaps related to the illness itself as it is to the antibiotics that patients received. And so that's really a sign that illness itself is driving changes in the fecal microbiome. And again, diversity seemed to play a role in outcome. Patients with less microbiome diversity stayed longer in the ICU than patients who had more diverse microbiomes, and length of stay was different. And so we saw some targets for potential probiotic intervention that we found many of them were common targets to what the IBD studies have found. And so we began to look at some of these as potential targets for probiotic therapy. And again, the changes in the admission ICU sample to the discharge sample seem to correlate with mortality, the changes in the microbiome's diversity. And so we'll have more data to come. We also know that perhaps we can use probiotic interventions like this to intervene on the cognition differences, anxiety, depression, and other changes that happen after illness or just with psychological illness themselves. And that's the idea that the gut microbiome makes neurotransmitters that affect the brain. And that's what we've had a whole new family of research come up called psychobiotics, a whole new family of potential interventions using probiotics to treat depression, anxiety, and other complications of neurologic and psychologic diseases. In fact, there's meta-analysis showing probiotics supplementation can reduce depression. And so this is an exciting future. So what's the clinical data for probiotic use overall in illness? So we know there's a great benefit for using probiotics to prevent antibiotic-associated diarrhea, which is common after antibiotics. This is a large study in JAMA, almost 12,000 patients, 40% reduction in antibiotic-associated diarrhea. And many different probiotics were successful at this. Again, giving back any fast-growing commensal organism, they will outgrow the pathogens like C. diff or other diarrhea-causing organisms very quickly and repopulate a healthy gut. Now, specific to ICU patients and pneumonia patients and the COVID-19 patients I care for who come in at risk for secondary bacterial infections, it really contributes to death and to complications in these patients. There was a study of Lactobacillus GG in patients who'd been intubated and placed on ventilators that was funded by the NIH, where they gave us a probiotic capsule with LGG to the mouth and one to the gut, and they found it reduced ventilator-associated pneumonia by half, 50%, published in a very high-impact critical care and respiratory journal. Again, the intention to treat analysis even showed a reduction by half of ventilator-associated pneumonias in these patients. C. diff, diarrhea, was also reduced as well. So again, this is a really meaningful study here showing benefits of giving probiotics in critically ill patients, potentially like those with COVID-19 that we're caring for now. There's new meta-analysis data my laboratory has done looking at all the trials of critical care probiotic interventions, and there's significant reductions in infections in multiple trials in thousands of patients. reductions in vanillin-associated pneumonia, and the sicker you were, the more beneficial the probiotics were, which makes sense because the gut is more deranged. The gut microbiome and other microbiome spaces like the mouth and the skin seem more deranged the sicker you are. And again, there was many studies involved in this and significant reductions in infections, again, from probiotics. That's also been shown in elective surgery, in sepsis, in trauma. There's many areas where probiotics have been shown to reduce infection and other clinical complications in many areas of medicine. And so there's a lot of promise here. But I think we needed a really landmark trial, and that has happened. For those of you who don't know about it, the landmark trial of a probiotic therapy was recently published in Nature, the very high impact, one of the premier journals in the world, gave 5 pages to a really important trial that looked at giving a lactobacillus bacterium plus a prebiotic in full-term infants in India, and then looking at infections and sepsis and death versus a control. And what they found in over 4,500 infants studied was that probiotics prevented sepsis and respiratory infections dramatically. This was a majorly significant effect, had a significant benefit on a combination of different infectious and even mortality outcomes. And so there was a significant reduction in the primary outcome combination of sepsis and death in the probiotic treated patients. And there were significant reductions in both culture positive or bacterial infections, but also culture negative, which were probably largely viral infections and other lower respiratory tract infections like potentially COVID-19. And so this gives us some promise, a great deal of promise, I would say, that we may be able to reduce COVID-19 and other viral infections using probiotics. And I think, like I said before, we in the Western world especially are set up for this in the industrialization of the world in general. More antibiotics, more processed foods, less fiber, more cleanliness to some degree, all sets us up for having a deranged dysbiotic bacterial flora, which reduces our ability to protect ourselves against viruses. The comorbidities that lead to greater death rates and severe illness rates from COVID-19, aging, diabetes, hypertension, obesity, all also derange the microbiome and set up those patients to be at greater risk from illness and death from COVID-19. And COVID-19 itself deranges the microbiome. We already have data that shows. So again, if we don't give probiotics to patients, they're going to be at greater risk. We know actually that the derangement of the gut microbiome increases ACE2 expression, which is how COVID-19 infects us, decreases inflammation, lung injury, and potentially severity of illness, we hypothesize. Giving probiotics like LGG, that I'll tell you about in the study we're doing soon, can restore that healthy gut flora. We think reduce ACE expression, reduce COVID-19 infection, and reduce severity. And that's our hypothesis that we think there's a lot of promise for. And there's already some evidence for this in healthy individuals, that giving probiotics can reduce upper respiratory tract infections, which are largely viral infections, by as much as 50%. And this is many trials, thousands of patients. So again, a strong signal of benefit for probiotics use in these kinds of infections. Reduces the severity of infections too, reduces the time of the infections by up to 2 days. So again, we have a real chance here to prevent infections. This was a larger meta-analysis, 10,000 patients, 16 randomized trials. The pre and probiotics can significantly reduce respiratory tract infections. So there's a lot of studies for other viral infections like this already. So we need clinical trials in COVID-19. And so we launched at Duke with Tony Sung's lab, my partner in this trial, the PROTECT EHC trial, which is probiotic use to eliminate COVID-19 in exposed household contacts. So again, we use this trial and identify patients or individuals who have acquired COVID-19, and then we contact their families, and we go treat everyone in the household from age 1 all the way up to age 100, at any age, with a probiotic to prevent others in the home becoming sick and perhaps getting a more severe disease. So we've published this protocol. It was just published about 2 weeks ago in British Medical Journal Open. So this is freely available online. We have a website for this if you want to learn more about this trial, protectehc.org. But again, you can learn all about the details of this trial in this free online paper. But essentially, this trial is all done online. We send the probiotic or the control to all of the family members of the different individuals we enroll. And again, we follow them for 28 days, and we collect stool samples at 3 time points and nasal swabs for COVID PCR. And then weekly, we get symptom checklists and any hospitalizations or other severity of illness scores from the patients all online. So the whole trial is done online, so anyone can enroll anywhere in the country, and we follow them out to day 60 for infection and their outcomes. So again, we've secured an IND for the lactobacillus GG we're using in this trial, and we've already enrolled hundreds of patients. And so again, we have a lot of questions we want to try to answer. One is, what is the risk of COVID-19 infection based on your baseline microbiome? And how do you get more severe disease if you have a more deranged or dysbiotic microbiome? And can probiotics treat certain people better than others and prevent COVID-19? So we think there's a lot of great answers to questions we can get about how probiotics affects our risk for viruses like COVID-19. So in closing, what have we learned so far? A healthy gut is a diverse gut, and that's what all of us should want. And probiotics can help us with that. ICU and other illnesses lead to significant loss of diversity of the gut and the mouth and other areas of the body associated with poor outcomes. We need to correct this, and using probiotics and other interventions like that can do that and allow us to change the world we live in. The world we live in being a microbial world, as we're largely microbial in our makeup, and potentially prevent and protect the world from COVID-19 with a safe, simple intervention like a probiotic. So I think the day is coming and is already perhaps here that we should be prescribing probiotics to our patients to prevent infection rather than antibiotics, perhaps, or in the place of antibiotics someday, and to treat and prevent other illnesses and prevent our risk of things like COVID-19. With the idea being we can resod the lawn that's been created by Western diets, by antibiotics, by critical illness, or COVID-19 itself, with probiotics, potentially stool transplants, and even poop pills. The kids at MIT will sell you their poop and tell you that it makes you smarter. FDA is curious about that. But nonetheless, I think there's great promise for probiotics to prevent illness and to protect against the severity of illness and restore balance to our microbiome the way it was intended when we as humans evolved. And with new technology and the simple internet clinical trials we can do now, we can make it so easy a child can do it. So with that, I'd like to thank Rob Knight and his lab for all the work they did on all the ICU trials we've done, and Tony Sung, my partner on the Protect DHC trial, and all the folks at Duke that are helping us. With that, I look forward to your questions. Feel free to follow me on Twitter and Instagram. We publish a lot of our new data on the microbiome and probiotics, and I share it on Twitter and Instagram regularly. And feel free to email me at the email below, and I'm happy to share any of these papers, this data with you. Thanks, and look forward to your questions.
Very interesting talk. There are already several questions. I think, one of my first question is, why are probiotics not being used more often in ICU? But it also relates probably to safety, because that was also the first question. You mentioned that probiotics are safe, but there's still this concern that maybe the harms are not published enough or not reported enough. And then there's a question to ask you whether you can comment on that, because I think safety is really key in ICU patients.
Sure. I think that's a great question. And I think the good news is now we have quite a number of trials of meaningful, high impact journal publications of well done, including NIH funded research trials in a broad range of ICU patients showing significant benefit, like, as I talked to you about the trial from Lee Morrow and his group in ventilator-associated pneumonia and the reduction of infections by 50%, reductions of ventilator-associated pneumonia by 50%. And they did not have adverse events in that trial. They have a nice description of some of the patients that they excluded. You know, if you had perhaps Gore-Tex grafts or other particular risks for probiotic administration, that paper does a very good job, and I encourage all of you to read it on some of the patients you might not want to give probiotics to, which is a very small window of patients that you might want to avoid them in. But for the vast majority of patients, that paper, and then many other papers that, you saw, I talked about the meta-analysis of thousands of patients and many, many trials where significant safety was seen and significant benefit in reduction of infections was seen. I really think this is moving to something that should be more and more routine in all of our practices to prevent infections like ventilator-associated pneumonia, which are devastating to our patients and are devastating complications when they occur, and C. diff, obviously. Again, there's a meta-analysis I didn't talk about in this lecture, but there's a meta-analysis of thousands of children and adults where you can reduce Clostridium difficile by over 60% in individuals. And so I think there's a really large now body of literature. And then, you saw the AHRQ in the US, a large FDA body, a large federal government body, looked at hundreds of trials, many of whom had acute illness, where they found safety and even benefit reductions of infections, not increased risk in patients who received it. I think the one caveat I'll make is don't give probiotics post-pylorically. Make sure if you're going to give them, either have them taken orally or have them given into a stomach feeding tube or an NG tube. The one trial that showed perhaps there was some risk from a pre-probiotic combination was a trial done where they were given to pancreatitis patients who, like Lead Ellis's, given in a post-pyloric into the small bowel where there was an ileus and the probiotic and fiber combination didn't move and caused some gut ischemia and issues there. So do not give them outside the stomach into the small bowel. I think that's a great lesson to learn. But given to the stomach, they seem to be very safe in the right kinds of patients. And clearly our data is showing more and more every day that they improve outcome.
Okay, that's a very great answer. I think there are several other questions on which probiotic to use and how to combine it with diets and also and now with for COVID-19, how to combine it with the vaccination, which is ongoing. But I also got a message that it's actually our time is already up. But Paul, you will receive all questions from the audience, and there's also still some networking opportunities. So I would like to thank you with this very positive but scientifically evidence-based presentation. So thank you very much.